My name is Marko Mutanen. I am holding a senior curator position in the University of Oulo. It may sound a bit weird, but I am actually nowadays not working in a museum, despite my title. I'm working in a research unit in the University of Oulo. My main group is Lepidoptera, butterflies and moths, but I'm increasingly also studying sawflies. I'm doing field work mostly in Finland, but in my research I'm not that strict. I'm doing through the collaboration research in a large geographic area, mostly Holarctic species. And I have been coordinating the barcoding activities through the FINBOL, the Finnish Barcode of Life initiative, since its establishment in 2011. And I have been the main responsible person for the barcoding of Finnish Lepidoptera and sawflies, Symphota. Okay, here is this FINBOL statistics shown. I'm not going to go through all these numbers. As you can see, it is about 63,000 sequences that we have generated so far of about 16,000 species. 3,000 less bins, and this difference is mostly because we have been quite also sequencing quite lots of fungi and plants that are not given bins. So that is for this difference. And actually we have been also, these numbers refer only to the records that are based on the specimen collected from Finland, but we haven't been so strict in that respect. We have barcoded actually quite many specimens collected abroad, but that are of species that are present in Finland. So actually we have barcoded quite many more species than that, Finnish species, and also in the neighboring countries, many Finnish species, especially in Norway and Germany, have been barcoded. So actually we have many more species barcoded elsewhere in total. I would say the 16,000 species is about half of the known species in Finland, and maybe about roughly about one third of the species that are expected to be present in Finland. Okay, so this FINBOL network was established 2010. We actually got funding. I earlier mentioned 2011. We got the first funds. Then it was established a bit earlier. The leading institution is my institution, the University of Oulo. The FINBOL is basically a researcher network. Only 3 institutions are involved. The 3 major museums of Helsinki, that is the main museum in Helsinki, and then University of Turku and University of Oulu. But also researchers from many other institutions are involved in FINBOL. Also many private or citizen scientists are involved. And presently we don't have a lot of funds, but we have got from many foundations and many sources from 2011 onwards. And by those funds we have been able to sequence quite many species. And we are right now waiting for the next decision. It is an application prepared together with these museums and also some other museums, and we are trying to get money. It's a bigger project. Barcoding is just one part of it, and we are already in the second round, so we are expecting to have the decision by the end of this year or early next year. And I would say that the chances to get that money appear quite high. It is an infrastructure project, so we have linked barcoding as a part of the Finnish Biodiversity Information Facility infrastructure project, and that way we are hoping to be able to barcode more in the future. Okay, I briefly present 3 different projects that are underway at the moment in Finland. The first project is an ecological study. It's actually a long study that started already a long time ago, and now incorporating also barcoding and meta-barcoding in the project. It is the project studies the ecology of boreal forest perch, the tit perch, and in this project the diet of the tits is to be clarified based on the meta-barcoding approaches. And the researchers want to understand in the boreal forests, the fauna is changing a lot at the moment because of the climate change. Many new species are coming and the northern species are retracting. So the fauna is changing fast, and they want to understand better how this affects to the birds' diets. And what is a novel aspect in that study that they are barcoding at the same time the bird droppings and the food availability, what is available. So the insect pellets, frost pellets are collected by these kind of funnels seen here, or even this kind of sealed seeds connected to the funnels. So this has been done for some years now, and that way one can get lots of those frost pellets of insects. So the idea is to meta-barcode those pellets and to say what is available, what are the caterpillars available in the trees, and it seems to work. Actually, we already have preliminary data. The system works. We can get a good idea what is living there in the trees, especially as in that area the reference library is very comprehensive. For example, for the Lepidoptera, it's complete. We can name just every single species that we get from there, and it is possible to get the barcodes also even from the insect frost to get an idea of them. The second project is a taxonomic project led by the citizen scientist Jukka Tabela, and the idea is to revise the moth genus Pleurota or Pleurotinae worldwide. They are mostly occurring in the Europe, southern Europe, and it is a badly known group of moths. Only about 50 species are so far described, but when Jukka started doing barcoding and collecting them in different countries, he already have described many new species and observed more than 100 bins. So that is an example of this kind of taxonomic project, badly known, many new species to come. And we have funded this project through the iBOL, so we haven't been very strict how we use the money that we have got. So we have funded also this kind of projects. And the third project is by Riikka Elo, who is working in the University of Turku. She is studying mites, soil mites, and those animals are not studied by many, but luckily we have some researchers. The soil fauna is very badly known. I think that is the same in most other countries, that very few people are working on them. So I'm very happy that Riikka has been active in mite barcoding, and she is going to continue that also in the future. So she has been sequencing to date about 1,000 specimens, if I remember correctly, and about 1/2 of the species that are occurring in Finland. And in that project, as you can see by this number, only less than 1/2 of the specimens worked for the sequencing, and we know that is not because of the small size of the mites, but because of the primer issues. So there is a lot to do to develop better primers. Riikka also wants to develop some alternative markers for the mites, even though barcodes are actually working very well. The divergences are very, very big generally in mites, so barcodes are working well. The CO1 barcodes, I mean. Okay, and then to the SWOT analysis. The strengths, we have lots of expertise in Finland, and that expertise is taxonomically very broad. So we have experts in most groups of insects as well. Of course, there are gaps, but we have very many people and expertise taxonomically broadly. We have very good collections, also private collections. Actually, in the iBOL we have always targeted also the private collections, and we have used them a lot because typically the specimens tend to be fresher, working better. So we have been working a lot with the private collections, but also with the museum collections. Our approach has been quite cost efficient. We haven't got very lots of funds compared to some other countries, but we have been doing quite cost efficiently, I would say. We have got quite extensive results. And the one strength in Finland is that the nation's economy is quite stable and strong, so there are opportunities to get funds for this kind of work. Also, the institutional commitment is of high level. The weaknesses, we have funders, as I mentioned, but we don't have very many really big funders, so it's not easy to get millions of euros for barcoding in Finland. There are some possibilities, but not many. And also the museums are not doing very well because of the cuts done by the government, so people even have been fired, so there are limited possibilities to provide in-kind contribution by the museums. Also, what we have learned from the previous projects is that our protocols have been too heterogeneous, so we have to streamline the protocols to get to be more efficient. And then to the threats, there is a threat, not very concrete and likely at the moment, but of course it may always happen that the key institutions don't give a priority to the DNA barcoding. So far we are in a happy situation, so the main museums are happy to support DNA barcoding. There is maybe this kind of threat that the situation changes in the future, hopefully not. And of course the main threat, we don't get funding in the future. Funding is the key thing, I think, always mentioned here. And also the fact is that to get and to be efficient in the future, it means that we have to go to the museum specimens, because it is increasingly difficult to get fresh samples because most species that are rare, and we don't have, it's difficult to get fresh samples of rare species, so we have to go to the museums and we have to start using the older museum specimens. And now luckily this technology has developed in that regards. The sequel, for example, works much better for the museum specimens, but we really don't know, we haven't tried at least, we don't have a very clear idea how well the museum specimens generally work. So that is a potential problem. And well, okay, of course maybe also negative turns may happen in the country's economy, and not very likely threat, but also that the users of the data don't find the barcodes very useful. I would say it doesn't seem to be so. I would say that the end users have been very happy to, and many other researchers are actually taking benefit of the very comprehensive barcode library that we already have. And then to the opportunities, the DNA barcoding is a very well known thing among the biologists. So I would say in Finland at least everybody knows what is DNA barcoding. Also iBOL and the FinBOL, they are very well known brands. So that means that it's easier to sell them for the funders because they are already so well-known brands. Also, the biodiversity crisis is increasingly recognized, and there are concerns when it comes to the biodiversity crisis and the climate change and these kind of things. So hopefully in the future these kind of things are supported increasingly. Then we are all hoping that the iBOL becomes a global infrastructure that is supported also at the very high political levels and by the countries, and that is what we all hope to see, of course. And as the DNA barcodes are increasingly used, we are hoping, of course, that there are those targeted calls by the Finnish Academy now and then, and we are hoping that at some point there will be a targeted call very suitable for the DNA barcoding as well. And also as the data comes bigger and bigger, it gives many opportunities to do mega analysis, very big data analysis that would be also a very good thing for the barcoding. Okay, that's all. Thank you.